at Triton Bio.
Great.
Lorenzo, see your screen, and go ahead.
Thanks, everybody. Right, so my name is Lorenzo D'Amico. I'm the founder of Triton Bio. And our company's mission is to ensure that patients with infections are treated with the right antibiotic at the right time. So why are we doing that? So actually, I'm sure most of you in the audience are now keenly aware of the fact that respiratory and other infections can be very dangerous and even deadly. What happens when infections start in these other body sites is that they can eventually, if they become serious enough, they can spread into the bloodstream and cause multi-organ failure. And so in the world we're living in today, this data is still kind of rolling in, but it's well known now that a major cause of death among COVID-19 patients is actually the secondary bacterial or fungal infection, which occurs and becomes systemic. But no matter what the cause of this systemic infection is, we know that sepsis, which is what the condition, the syndrome is called when an infection spreads and triggers an immune response in the patient, is very, very dangerous and deadly. Not only that, this represents one of the most costly conditions to treat, at least in the United States. So why is this such a dangerous problem? Why is this such a serious problem in society? And it really comes back to how the treatment plan is guided by laboratory medicine. So our primary customer is the lab director. The lab's job is to take samples from patients and determine what the cause of the infection is, identify that organism, and determine which antibiotics can be used to treat that infection most effectively. And so the lab would report a chart similar to the one that I'm showing here at the right, which determines the organism's identity and its resistance to different types of drugs. And the issue here is that the lab process takes anywhere between 2 to 5 days for most common bacterial pathogens. The time within which physicians must treat patients is really on the order of hours, which is what this table is showing, this chart is showing at the right. And so really the lab turnaround time is far too long to effectively guide therapy. That means that the doc left with what they call empiric therapy, which is actually a kind of guess and check with antibiotics to determine how really they're guided by how a patient is responding to a drug. That has a number of important consequences that I'm not going to enumerate here, but suffice it is to say patients' lives are jeopardized when they are not treated with the right drug. And so our goal as a company, we've invented this technique that we call cellular chromatography. We're going to replace these slow, outdated culture techniques with an automated instrument system that uses a pretty low-cost cartridge. And the target that we're looking to do is to deliver that same report that the clinical microbiology lab is delivering today, specifically identifying the organism and determining quantitatively its drug resistance profile. And we're targeting a 30 to 60 minute turnaround time per sample. The system works briefly is a strategy wherein a liquid sample is processed through a proprietary fluidic processing unit that uses a combination of physical forces to segregate mixtures of bacteria, fungi, and any host material that comes in the sample, like blood cells or epithelial cells in the case of a lung sample. And by segregating those organisms out, what we're doing is we're measuring a unique fingerprint of each organism, and we can monitor how that fingerprint changes in response to in vitro challenge with an antibiotic. So why does this product make sense in the laboratory setting for hospitalized patients? So we've conducted several hundred interviews with key stakeholders in this industry. And what we've come to understand is that the pressures that these different stakeholders face all really must be addressed if a product is to be adopted. And so when we think about the primary customer, the lab director, the challenge really here is about the hands-on time that their technicians have to allocate to manually process these samples. When you look at clinical staff, of course, the clinical problem is really about turnaround time, the time to the result. But very importantly, we discovered while on the road before COVID. The hospital administrators, the folks who have to really balance the budget, think of their microbiology labs as cost centers. And so what we understood in that was that when we saw products that were being bought today, it was really about enabling the labs to capture more reimbursement value. And so what I mean by that, when we look at a comparative sort of pricing model where the conventional method is here on the left and our method, our combo method, which eliminates culture, is shown at the right, we believe that for a typical hospital laboratory, which will process about 5,000 samples a year, just jumping to the punchline here at the bottom of that chart, we think that labs can capture about $70,000 more in profit by performing these laboratory tests with our instrument. Now, how do we make money? So our strategy is to be an asset sale. And so there's a one-time large cost of $200,000. And then there would be recurring revenue. Specifically, there'd be a license to the database. So those fingerprints of different organisms, there'd be an annual license to that and a maintenance contract to service the machine every year. And in addition, there's a relatively low cost of ownership in terms of running each individual test cartridge. And so we estimate the lifetime value of these lab directors to be about 8 years, and the value to be about $320,000. What that would mean for the labs is they have a payback time on our machine of about 4 years. This is separating out what some of our competitors try to argue, which is sort of the soft cost saving of giving patients the right antibiotic earlier may lead to earlier discharge, shorter length of stay in the ICU. Definitely a problem that is top of mind for hospitals today in the middle of this COVID crisis. But we think that the stronger argument to be made for adoption is really on that hard cost benefit. So think about our target market. We think these value propositions really make more sense for hospital labs where lab turnaround time is still an important driver. When you look out at really what we're competing with are these automated Petri dish culture systems. Their primary customers are going to be places like Quest and LabCorp that are mostly doing urinary tract infection testing. They're going to mostly be negative samples. Time is not really important in those settings. But in the context of bloodstream infections, in the ER, in the hospitalized patients, ICUs, we think that the hospital labs make the most sense. And so in the United States, where we have more reliable data, this looks like almost a $3 billion market opportunity, but our beachhead, we believe, is going to be among these top, more prestigious academic medical centers. Our intelligence about the market indicates that these places are really early adopters. They like to be ahead of the game. A large driver for those lab directors is being authors on papers and earning fees, honoraria, that was the word I was looking for, to speak on behalf of companies. So we think of those as early adopters. So how we plan to get there? Right now, the company has an SBIR Phase 1 award from the NSF. We also have a research contract partnership really with the Baylor College of Medicine, where we are a spin out of. We're using that money right now to complete prototyping of our instrument and to generate preliminary data. And is to do a $1 million seed to expand our proprietary database and really demonstrate proof of concept, which could go into a RUO product to continue to expand that that knowledge database, which is that database that we would license. Some important comparable companies that were acquired in the last several years, I've shown here at the top right, at the top of the chart. But the plan really is to try to get in the market in about 5 years with our first version of this system. So a bit about myself, I recently left the academy. I've led a number of research grants while I was at the Baylor College of Medicine. I have a fantastic team of advisors. Peter Gascoigne was my PhD mentor. He's got 40 years of experience in the core technology that this system's built on. Jill is our business advisor. She's spent the larger part of her career really leading mergers, acquisitions, licensing deals among major diagnostics companies. And then Dr. Ajami, who is a co-founder of the company, he has a lot of expertise in really microbiome research and how we applied this technology to microbiome problems while we were funded by DARPA. And so yeah, our next step, so we're an LA-based company. We work out of the Lundquist Institute, the biolabs incubator there. Immediate next steps are to continue to build out our team with our seed money, and so attract some talented folks to help us complete the prototype and that initial database, as well as complete international patent filings. With that, yeah, happy to take questions. Thanks for your attention.
Mimi, do you have any questions first?
Yes. So can you tell me about the technology in which was started and how that came about?
Yeah, so happy to. The earlier versions of the system were used to isolate circulating tumor cells from blood. What the system does is it's actually a microfluidic and microelectronic device. So the liquid sample would flow into the device, and these microelectrodes, which are embedded in the channel, are able to exploit electrical biomarkers of cells. So they're sort of intrinsic physical properties of cells. In the case of bacteria, the forces that we apply on them are unique because they all have sort of unique, at a species level, they have unique electrical characteristics, which is a result of their bacterial cell wall.
So how is that? Is that a patent that, I mean, are you licensing this technology from university, from Baylor? Or are you, I mean, how is this defensible in a patent?
Sure, yeah. So we have one patent application, which is we're about to file in abroad in EPO that is owned by the company. And we have a term sheet from Baylor for that cytophysical profiling technique I'm describing, as well as some other hardware, sort of an apparatus and a method patent, which we're working with them to. I'd be happy to share that and get your opinion on it. I don't think the terms are super great yet, but I think that we can get the college to come around. Yeah.
I'm sorry, you're breaking out. I just had a motorcycle drive by.
Oh, I'm sorry. Yeah, what I was saying is that the college owns the patent to this. It's not a patent application yet. The invention for the cytophysical profiling, and so. We are currently negotiating a term sheet with them. I'd be happy to show it to you. I've gotten some other folks' input on that term sheet. I don't think it's great. And so we're trying to work with the college to come to more favorable terms before we execute the agreement.
And who's your attorney?
I was going to say, before we get to the next judge questions, I just want to let everyone know that we're just going to go over a little bit more time, but please stay on, because we do have an Audience Choice Award for the teams, and if you can stay on so that we can go through that voting, that would be awesome. But we've only got a couple more questions to go. Please bear with us. Mimi, did you have another question, or can I move on to Lucas?
Yes, I see the floor to Lucas.
Okay, all right.
Thanks, Mary. So I saw your team. I see a lot of advisors. I was a little curious. Is it just you as a solo founder right now with a strong team of advisors, or who else is on the team?
And, I'm the solo founder at the moment.
Okay. Go to market. I feel like I saw a lot of technology pieces. I want to understand how you're actually going to attack these labs and get the product in there, whether you have the prototype or not.
Yeah, it's a great question. I think in the early days, I think this has to be a venture-backed thing and this is true for many diagnostics. I don't think that you get strong adoption until the data is very extensive and reliable. That's just a limitation of FDA. These are things that are required, which is why the RUO method isn't. So the strategy there really, I believe it to be really direct sales, relying heavily on partnerships to generate buzz in the scientific literature. So publishing papers with beta test sites in reputable journals, I think is a first step towards that effort. And in that situation, in that scenario, you know, these machines are probably placed for free. I don't imagine there'd be strong incentive to buy initially, right? If the lab can't get paid to use it with an FDA clearance. And authorization to get reimbursed, which is a secondary challenge after FDA clearance. So that's the strategy really in the first phase, is to try to get early adopters through research use only.
And I might have missed it, but have you received any grant rounds?
Right. Yeah. So we have CIR one National Science Foundation. The core technology was developed by a DARPA grant when I was still working at Baylor, and then we also have a contract with Baylor. It's kind of like a sub-award to continue developing this technology. So also grant. So we have all non-dilutive funding at the moment.
Awesome. Appreciate it. Next presentation.
Thanks.
Arye.
Yeah, I'll be really quick. Hey, Lorenzo. So Lorenzo and I have talked before, so I've already got the inside scoop. So I'm curious how you arrived at the price of the instrument, $200K.
Yeah, you know, the way I calculated that was really based on what I thought was an attractive payback time based off of that reimbursement gain, as well as what I think is realistically the throughput of the system. So if a single instrument can do about 30 samples a day, I worked kind of backwards on that. Also thinking about comparable technologies. So I think of this system in terms of what it's sort of capable of doing is there's a machine that's already in the market called the Bruker Biotyper. It's a mass spec multi-TOF system. And their price is also about $200,000. I think of it as a very similar business model. They argue of like a 3 to 4 year payback time. And that, Arye, when I went around the country and was interviewing folks, that was everybody's favorite machine. People loved the clear-cut ROI, the ease of use. Those were things that really resonated with folks. And so that's how I arrived at that $200,000 price. I definitely welcome pushback on it. You know, it's early stage, still thinking through these strategies. But it felt like a realistic target.
Okay, great. Yeah, no further questions for me.
Cool. Thank you.
All right. Thank you, judges.